Hi, I'm Chippy, and this is Ethics Unfiltered, where we talk about science, innovation, and how we protect people from bad ideas in good packaging. This series is called The Ethics of Innovation, and in it, we're going to lay down the groundwork for understanding how ethical science works, how regulations were born, and how to tell the difference between real research and pitch deck methodology. Because if you're building in health or biotech, or just following the conversation, you deserve to know when something's promising and when it's pretending. Let me say this up front. I am not anti innovation. I love good ideas. I love breakthroughs. I love when people try to do something that's never been done before. But what I am against is taking shortcuts, especially in medicine and biology, where shortcuts don't just create bugs or bad press, they create harm. And these days, a lot of startups, especially the ones dipping their toes into healthcare and biotech, are acting like the rules don't apply to them. Like ethics is a formality, like oversight is a blocker, like science is just a word you sprinkle into your deck to sound credible. And honestly, that's dangerous. Because here's the thing, not every sciencey project is research. Not every product launch with biometrics and charts is a study. Not everything that sounds impressive means anything. So today, we're going back to basics. Before we talk about ethics boards or regulations or peer review or any of the reasons those exist, we are going to talk about what actually counts as research. Because if you're going to ask whether something is ethical, the first thing you should ask is it even research in the first place? So, what is biomedical research? In plain terms, biomedical research is a structured, data driven process for answering a specific question about human, animal, or biological health. That might sound obvious, but it matters. Research is systematic, it follows a plan, it asks a question, it collects data in a way that's consistent, reproducible, and transparent. And it's designed to create knowledge, not just a product. This is different from development. It's different from testing a prototype. And it's very different from doing something cool once and writing a Medium post about it. So if someone says, we tried this out on a few people and they felt better, that's interesting, maybe even promising. But it's not research yet. Because research doesn't just report outcomes, it investigates them carefully, repeatedly, and with enough structure that someone else can do the same thing and compare their results to yours. Let's talk about research goals and SMART objectives. A real study starts with a question that's specific, measurable, achievable, relevant, and time bound. That's what SMART stands for. If your study goal is something like, we aim to optimize human potential and cellular longevity through personalized feedback systems. Okay, sounds nice, sounds aspirational. But what are you actually measuring? What's the intervention? Who is involved? And over what time span? So that's not a SMART objective. That's a TED Talk thesis. A real example might sound more like, we want to know whether starting treatment early for patients with ocular hypertension reduces the risk of developing glaucoma over 5 years? That's a real study question. It's testable, it's concrete, it has a clear population, intervention, comparison, and timeline. Without those things, you don't have a research question, you have branding. And then we need a method. This is where good science shows its work. A solid study design will usually include a control group, so you know what happens without the intervention, an intervention group, so you can compare results, randomization when appropriate to reduce bias in who gets what, a sample size calculation to make sure your data isn't just coincidence, defined endpoints so you know what success or failure actually looks like, and a statistical plan decided before you collect the data. That last one is important. If you start analyzing data only after you've seen the results, it's really easy to find something that looks meaningful but actually isn't. That's called data dredging or p-hacking, and it's one of the fastest ways to end up publishing a discovery that's really just a coincidence. But let's zoom out for a second, because all of these pieces, your study design, your endpoints, your stats plan, they should all be written down in a research protocol before you ever touch a subject or collect a single data point. The protocol is your blueprint. It says, here's what we're doing, here's how, and here's why. And in real research, that protocol goes through peer review and ethics review before the study starts, not after, not next week. So, when a company says we have 500 times more data than anyone else and we're submitting it for peer review next week, that's not impressive. That's a red flag. Because what they're telling you is we did the science already, but we're just now getting around to asking if we did it right. That's backwards. You can't retroactively call something ethical and rigorous after it's already done, especially when it involves real people, real animals, or real consequences. You don't design your research protocol after you've already designed your dog. Let's talk about outcomes. Because what you choose to measure tells me everything I need to know about whether this is science or just fancy branding. In medicine, we don't always have the luxury of waiting 10 years to see if someone has a heart attack, goes blind, or develops dementia. So we use what are called surrogate markers, or shortcuts that reliably predict what's coming down the line. Let's take examples most of you will know. Fasting blood sugar predicts risk for diabetes. Blood pressure tells you about your heart health and your stroke risk. Cholesterol levels are strongly linked to cardiovascular outcomes. These aren't vague numbers made up for a PowerPoint. These are validated, peer-reviewed, clinically meaningful indicators. They've been tested across thousands of patients, and we know what they mean, how to interpret them, and when to intervene. But when you start seeing things like mitochondrial resilience index or bioenergetic optimization score, we have to pause and ask, what's being measured here? How is it validated? Can someone outside your company reproduce it? Because if only your platform knows how to define or interpret the outcome, it's not a scientific metric. It's a branded experience with scientific vibes. And that's okay if you're transparent about it. But please don't pitch it like it's a clinical grade endpoint. A quick word about jargon. While we're here, let's talk about language. Yes, medicine uses a lot of jargon. But here's the thing: biomedical jargon is standardized. That's the point. If I say intraocular pressure and someone in Argentina or Denmark or Canada says intraocular pressure, we're all talking about the same thing. That's not about gatekeeping, it's about clarity. This is something we learn early in medical training, especially in problem based learning models like the Maastricht 7 jumps. Jump 1 is clarify the terms. You can't even begin a discussion unless you're sure you're talking about the same thing. So, yeah, it might take a second to look something up, but medical terms are built for consistency, not confusion. In contrast, a lot of tech speak uses language that's intentionally vague. It sounds biomedical, but when you try to pin it down, it slips away. We'll unpack this more in a later episode. But for now, just know if you don't understand what a term means, it's not because you're not smart enough. It might be because it wasn't actually designed to mean anything. Okay, you've made it this far. You know what research should look like. Let's talk about what to watch out for when it doesn't. Here are a few red flags in startup research. There's no mention of an independent ethics board or institutional review board, just vague hand waving about internal ethical frameworks. There's no control group, no sample size justification, and no explanation for why they skipped those steps. Claims of effectiveness are based on tiny pilot runs, and results are being shared in press releases or podcast interviews before any peer reviewed study is published. Methods are proprietary and can't be explained. We'll publish soon, but it's been 6 months and all you've seen is a YouTube demo with motion graphics. And again, not every study will be huge, but if they're making big claims, asking for money, or positioning themselves as scientific leaders, these are fair questions. And if the answers are vague, defensive, or evasive, you're allowed to be skeptical. In fact, you should be. So, what can you do? You don't need a PhD to ask better questions. In fact, some of the best questions are the simplest. What was the sample size? Was there a control group? Was it randomized if they had intervention like medicines or medical devices? Was it peer reviewed? Can I see the data? And here's the truth that often gets lost in all the jargon and PR gloss. Real researchers welcome questions. If someone's done the work carefully and ethically, they're usually excited to explain it. They'll walk you through their methods, their data, and their reasoning. And if they can't talk about something yet, because it's still under review or the manuscript isn't out, that's okay too. But they'll tell you why. They'll say, we're still in the ethics approval process, or it's an ongoing study. We'll share more once it's peer reviewed. What they won't say is, you just don't understand the science, or trust us, we'll explain later. Because that's not science, that's salesmanship. So, if someone makes you feel silly for asking basic questions, or acts like your curiosity is an inconvenience, that's a red flag too. Science doesn't require blind trust, it requires transparency. And the more people who know how to look under the hood, the stronger our innovation and our ethics become. This might all sound a little picky, a little academic, but it's not. It's the foundation of doing science responsibly. Because when research gets sloppy or vague or unreviewed, people get hurt. Bad studies can lead to bad products, bad policies, bad health outcomes. And it doesn't matter how well intentioned the people were if the method was broken from the start. This is why we have ethics boards. This is why we have regulations. This is why we don't just move fast and break things when people's bodies, minds, and lives are involved. In the next few episodes, we're going to look into why these standards exist, where the rules came from, what happens when they're ignored, and how some of the most tragic moments in science led to the protections we have today. We'll talk about the Nuremberg Code, the Belmont Report, IRBs, animal research guidelines, and why moving fast in science sometimes means running straight into the same mistakes we already promised not to make again. Thanks for listening, and welcome to Ethics Unfiltered.